Photosynthesis is made up of two sets of reactions, the light reactions or light-dependent reactions, which power the dark reactions, light-independent reactions, or the Calvin cycle. As you can see here in this slide, light reactions occur in the grana of the chloroplast, and the dark reactions, Calvin cycle, or the light-independent reactions occur in the stroma of the chloroplast. As in cellular respiration, photosynthesis requires electrons, but for the opposite reason. In photosynthesis, the electrons are needed to build up sugars. In cellular respiration, electrons are needed to transfer from one molecule to the next to drive redox reactions, which drives the production of ATP ultimately. In the photosynthesis, the source of electrons is water. In cellular respiration, the source of electrons is glucose. So we have opposite reactions going on. However, in photosynthesis, to get these electrons from water moving, light energy is needed. The two steps, as mentioned before, of photosynthesis are the light-dependent reactions and the light-independent reactions. We'll first discuss the light-dependent reactions, which are driven by light striking the pigments, and there's two parts of the light-dependent reactions, the cyclic electron flow and the non-cyclic electron flow. As light passes through a leaf into a cell, and specifically into a chloroplast, that light transmits through the outer membranes of the chloroplast and strikes the membranes of the thylakoid membranes. This light is absorbed, and of course chlorophyll A and B reflect the green light. It is in the thylakoid membrane that photosystems, which are made up of pigments, the primary pigment being chlorophyll A or B, and accessory pigments. This is the location for the transmission of energy from light to chemical energy. Here in this slide shows the typical structure of a photosystem, which, as you recall, is located in the membrane of the thylakoid. Here we have light or photons of light striking the accessory or antenna pigments, which relay that light energy to the reaction center, which is made up of chlorophyll A or chlorophyll B. It is here at the reaction center that the electrons are excited to a higher energy level and passed on to what you call a primary electron acceptor, which goes on to drive the two stages of light-dependent reactions. This slide, which will be shown again later in this podcast, shows a general overview of the flow of electrons from water through non-cyclic electron flow to NADP, which picks up those electrons and is reduced to NADPH. Both products of the light reactions, NADPH and ATP, go on to drive the Calvin cycle. But first, let's discuss cyclic electron flow. Hence its name cyclic, meaning it goes around and around. The electrons in cyclic electron flow never really leave the thylakoid membrane. Cyclic electron flow generates a chemiosmotic gradient between the thylakoid space and the stroma. This chemiosmotic gradient then allows for protons or hydrogens to flow down their concentration gradient into the stroma through the enzyme ATP synthase, which drives the production of ATP. The same process occurs in cellular respiration, however the structures are different. But more on this later. So let's get to the cyclic electron flow. Photosystem 1 is the only photosystem that is responsible for cyclic electron flow. Here, as you can see, the vertical region of the slide represents the amount of relative energy. The electrons are excited in photosystem 1 by a process called photoexcitation. These electrons are excited and are picked up by a primary electron acceptor in the membrane and pass through the electron transport chain, which is of course made up of cytochrome complexes. The flow of the electrons powers the proton pumps, which generates a chemiosmotic gradient, which drives the synthesis of ATP. Same exact way is done in cellular respiration. Now let's discuss non-cyclic electron flow. Non-cyclic electron flow means the electrons that start this flow do not end up where they started out. This non-cyclic electron flow creates a chemiosmotic gradient just as in cyclic electron flow. However, the ultimate end location for the electrons is different. First, we get photoexcitation again, but however, this time it's photosystem 2. The electrons are excited by light in photosystem 2's reaction center, where they're picked up by a primary electron acceptor. After the electrons are lost by photosystem 2, water is then split by enzymes, and the electrons from the splitting of this water, or photolysis, are replaced into photosystem 2. Then these electrons that were excited by the light in photosystem 2 travel through the electron transport chain, which again drives the chemiosmotic gradient, which drives the synthesis of ATP. As you notice, as the electrons move from the primary electron acceptor down the electron transport chain, they will G. Once the electrons end up on photosystem 1, they get excited again by light, photoexcitation, to where they're picked up by another primary electron acceptor. As you can see, the energy of this process is even higher. From here, those electrons go through another electron transport chain where they finally are picked up by NADP reductase, which is an enzyme, as it's named, reduces NADP. This enzyme gives the electrons to NADP+, and NADP+ pick up hydrogens. As you can see again in this slide, it's a summary of non-cyclic electron flow. But you must note to remember, non-cyclic electron flow takes electrons ultimately from the splitting of water, drives those electrons passing through the electron transport chain, drives the production of ATP synthesis, and then end up on photosystem 1, and then those electrons end up on NADPH. So the final products of the light-dependent reactions and non-cyclic electron flow are NADPH and ATP. These products will go on to drive Calvin cycle. Great. Now, ATP, NADPH is two of the products of the light reactions, and oxygen is the product of the splitting of water in the light reactions. And if there's carbon dioxide present, what happens next? Well, there is this wonderful little enzyme called ribisco or RUBB carboxylase, one of the most important enzymes on the face of the earth. So, the first part of the Calvin cycle requires the fixing of carbon dioxide onto a 5-carbon molecule, sugar molecule called RUBP or ribulose biphosphate. This RUBP has 2 phosphate groups on either end. The enzyme that fixes these carbon dioxide molecules onto RUBP is called ribisco, like mentioned before. When this happens, 6-carbon intermediates, unstable intermediates, are created and immediately cut in half into 2 3-carbon molecules. These 3-carbon molecules are then phosphorylated by the ATP created in light-dependent reactions into a different form, and then these molecules are reduced by NADPH as a product of the light reactions, and to a molecule by the name of glyceraldehyde 3-phosphate or G3P. And finally, of those molecules, 2 of them are recycled back into RUBP by ATP. This slide shows 3 carbon dioxides being fed into the Calvin cycle and 3 RUBPs. This is because it takes 3 carbon dioxides to create 1 net gain of a 3-carbon sugar. However, as you know, glucose has 6 carbons, and so 6 carbon dioxides are needed. So this slide would turn twice, or the Calvin cycle would turn 6 times to generate 1 net gain of sugar or glucose. The remaining molecules are then recycled back into RUBP, which are needed for more Calvin cycle reactions. So, to summarize the dark reactions, or Calvin cycle, or light-independent reactions, you must know the reactants and the products. What goes in are carbon dioxides, RUBP, ATP, and NADPHs. And what comes out are glucose, and again RUBPs, ADPs, and NADP pluses. The ADPs and NADPs go back to the light reactions to be re-energized or reduced so they can come back to drive the Calvin cycle. Again, this is showing you, the slide is showing you the products of the light reactions drive the Calvin cycle with the addition of carbon dioxide.